If we were to look at your grandparents and your parents and yourself, we could assess how you inherited many of your traits and therefore could make predictions as to the combinations of genes that you carry. We also might be able to make some predictions as to what genes and therefore traits you might pass down to your own offspring. These predictions can be outlined by drawing Punnett squares. We will now study how genes are transmitted to offspring, so let's review the generations through which these genes are passed. The P1 generation is referred to as the parental generation, and the parental generation gives rise to the F1 generation, which is the first filial generation. Filial, once again, is derived from the Latin word for child. The F1 generation then gives rise to the F2 generation, which is the second filial generation. If we take a look at 3 generations, really what this means is we start with grandparents, we end up with parents, and then finally with children. A Punnett square is a chart used to predict the genotypes and phenotypes of offspring from a cross. A monohybrid cross is a cross of 2 individuals that are heterozygous for one gene. Mono means one, referring to the one gene, while hybrid means mixed. This refers to the heterozygous parents having one dominant and one recessive allele. The example we're going to look at is crossing 2 pea plants that are heterozygous for the seed color gene. In the P1 generation, pea plants that are true breeding for the dominant yellow phenotype are crossed with true breeding plants for the recessive green phenotype. This cross produces F1 heterozygotes with a yellow phenotype. Punnett square analysis can be used to predict the genotypes and therefore the phenotypes of the F2 generation. If you take a look at the F1 heterozygotes, the gametes receive either a dominant or a recessive allele. If we put the male gametes across the top of the Punnett square and the female gametes down the side of the Punnett square, there's 4 possibilities in the offspring. If a sperm with a dominant allele fertilizes an egg with a dominant allele, the offspring will be homozygous dominant and have yellow seeds. If a sperm with a dominant allele fertilizes an egg with a recessive allele, the offspring will be heterozygous and have yellow seeds. If a sperm with a recessive allele fertilizes an egg with a dominant allele, the offspring will be heterozygous and have yellow seeds. If a sperm with a recessive allele fertilizes an egg with a recessive allele, the offspring will be homozygous recessive and have green seeds. If we take a look at the ratios below, we can see that the genotypic ratio is 1 to 2 to 1. This corresponds with 1 of the 4 offspring being homozygous dominant, 2 of the 4 offspring being heterozygous, and 1 of the 4 offspring being homozygous recessive. This then corresponds to a phenotypic ratio of 3 to 1, meaning that 3 of the 4 offspring have dominant yellow seeds, and 1 of the 4 offspring have recessive green seeds. So as we saw in the previous Punnett square, a monohybrid cross has 4 possible offspring. The expected genotypic ratio is 1 to 2 to 1, corresponding with 1 homozygous dominant to 2 heterozygous to 1 homozygous recessive individuals. The expected phenotypic ratio is 3 to 1, corresponding with 3 of the 4 offspring having yellow seeds, which is the dominant trait, and 1 of the 4 offsprings having green seeds, which is the recessive trait. Mendel did similar crosses to study other pea plant traits as well, such as flower color, which we reviewed in Part A of this lecture, seed shape, pod color, pod shape, and height. As we saw in the monohybrid Punnett cross, and as we learned in meiosis, each gamete, meaning an egg or sperm cell, receives only 1 allele for each gene because it receives only 1 member of a homologous pair of chromosomes. This leads us to the law of segregation that states that 2 alleles of a gene separate from one another during meiosis. This is important because when a sperm fertilizes an egg, the offspring receives 1 allele from mom and 1 from dad. Homozygous individuals have 2 copies of the same allele for a given gene, so all of the gametes from this individual will have the same allele for that gene. However, heterozygous individuals have 2 different alleles for a given gene, so half the gametes contain the dominant allele and the other half contain the recessive allele. During meiosis, gametes are generated that contain one member of each homologous pair of chromosomes, and therefore one copy of each gene in a pair. 50% of the gametes will receive one of the two chromosomes and therefore one of the two genes, while the other 50% will receive the second chromosome and the second gene in the pair. Humans have 23 pairs of chromosomes, but here we are tracking only one pair. The chromosomes and therefore the alleles separate into the gametes that then fuse during fertilization, giving the offspring one allele from mom and one allele from dad. Mendel, of course, studied pea plants, which is a good model organism, but we are most interested in the transmission of traits in humans. So does the inheritance of traits in humans follow the same patterns as in pea plants? The answer is a resounding yes. The inheritance of over 5,000 human traits have since been shown to follow Mendel's patterns of inheritance. One example is the inheritance of freckles. The inheritance of freckles in humans follows the same transmission probabilities that were seen in pea plants for seed color. A monohybrid cross between two heterozygous parents leads to a 1:2:1 genotypic ratio and a 3:1 phenotypic ratio in the offspring. We just studied the transmission of one trait. However, how are two traits inherited? Mendel studied this to answer the question, does inheritance of one trait affect inheritance of a second trait? Mendel went on to perform dihybrid crosses. A dihybrid cross is a cross of two individuals that are heterozygous for two genes. Di means two, so in this case referring to two genes. Mendel studied seed type in pea plants. This includes seed color and seed shape. Seed color includes yellow or green, where yellow is dominant. Therefore, we use an uppercase letter Y for yellow and a lowercase letter y for green. You may wonder why we don't use a G for green, but by using the same letter, Y, and having one uppercase and one lowercase, we immediately know that this refers to different alleles of the same gene. Seed shape is either round or wrinkled, where round is dominant, so round is indicated by a capital R, and wrinkled is indicated by a lowercase r. This dihybrid cross of pea plants involves the genes for seed color and shape. Every offspring that has a dominant allele will have the dominant trait corresponding with that allele. Notice that there is a mix of all traits in the offspring. While the genotypic ratio is complicated to calculate, the phenotypic ratio from a dihybrid cross between two heterozygous parents is always 9:3 to 3:1. From the Punnett square on the previous slide, we saw that a dihybrid cross had 16 possible offspring with an expected phenotypic ratio of 9:3 to 3:1. This equates to 9 of the 16 offspring having yellow round seeds, 3 of the 16 offspring having yellow wrinkled seeds, 3 of the 16 offspring having green round seeds, and 1 of the 16 offspring having green and wrinkled seeds. Another way to state this is that 9 of the 16 offspring are expected to have both dominant traits, 3 of the 16 offspring are expected to have one dominant and one recessive trait, while 3 of the 16 offspring are expected to have the other recessive with the other dominant trait, and only 1 of the 16 offspring are expected to have both recessive traits. This puts in perspective just how rare recessive traits are. Inheritance of two traits in humans, in this case freckles and widow's peak hairline, follow the same transmission probabilities that were seen in pea plants for seed color and shape. A dihybrid cross between two heterozygous parents leads to the same 9:3 to 3:1 phenotypic ratio in the offspring. The results of Mendel's dihybrid crosses lead us to the law of independent assortment. This states that alleles of one gene on one chromosome distribute to the gametes independently of the alleles for a second gene on a second chromosome. For example, the seed shape alleles on one chromosome had no effect on seed color alleles on a second chromosome. This results in gametes with any combination of dominant and recessive alleles, and therefore offspring with any combination of dominant and recessive traits. For example, round and yellow or round and green seeds. This is similar to humans. While While we may expect that an individual with brown hair will also have brown eyes, it is possible for a human to have brown hair with blue eyes. This means that the inheritance of brown hair color has no influence on the inheritance of eye color. Random or independent assortment during meiosis can be demonstrated by considering a cell with a set of 2 chromosomes. In this case, there are 2 possible arrangements at the middle of the cell during metaphase. In this first scenario, both blue and both red chromosomes line up on the same side of the cell and end up in the gametes together. However, there's a second scenario on the next page. In this second scenario, the blue and red chromosomes line up on opposite sides of the cell and end up in the gametes together. In this example, there are 4 possible genetic combinations for the gametes. What this means overall is that the inheritance of one blue chromosome has no effect on inheritance of the second blue chromosome. Therefore, any mix of blue and red chromosomes, and therefore any mix of the genes on those chromosomes, can end up in the gametes and ultimately in the offspring. This is what the law of independent assortment states. Linked genes refer to alleles that are on the same chromosome. Alleles on the same chromosome typically can't separate from one another to go into different gametes, so they are usually inherited together. For this reason, these genes don't follow the law of independent assortment that we just discussed. These are linked genes since they are on the same chromosome. Therefore, they will most often end up in the same gamete and will be inherited together in offspring. As we saw, a Punnett square for a dihybrid cross following 2 genes had 16 possibilities for the offspring. Therefore, Punnett squares for more than 2 genes are much too large to draw. Therefore, the product rule can be used instead. The product rule states that the chance that 2 events will occur together equals the product of the chances of the single events occurring separately. This shows you how the product rule is used. The forked line method can be used to analyze a trihybrid cross, which would follow transmission of 3 traits. Here, the probability for seed color in the F2 generation occupies the top row, 3 yellow to 1 green. The probability for seed shape occupies the second row, 3 round to 1 wrinkled. And the probability for height occupies the third row, 3 tall to 1 dwarf. The probability for each possible combination of traits is calculated by multiplying the probability for each individual trait. Thus, the probability of F2 offspring having yellow, round, and tall traits is 3 times 3 times 3, or 27. Don't panic. You do not need to know how to do this math for your upcoming exam.